Good morning. My name is Anis Karuniawati. I am from the Department of Microbiology, Medical Faculty. The learning objective of my lecture today is about molecular pathogenesis of infection. To learn the pathogenesis, of course, we have first to learn host-parasite relationship, and I will focus in basic concept of infection. As we know that the disease will be produced by three main components. They are host, agents, and environment. When the three of them are in balance, then there are no disease. But when one of them or two of them are imbalanced, then the disease will appear. When we talk specifically about infection or infectious disease, we have to understand about Koch's postulate, which is stated by Robert Koch in 1890, and the postulates contain of four important things. One is that the microbe must be present in every case of the disease. The second one, the microbe must be isolated from the disease host and grown in pure culture. Means that we have to prove it that the microbe is there, that the microbe is in the specimen of the patient. The third postulate is the disease must be reproduced when a pure culture is introduced into a non-diseased susceptible host. Means that if we get the isolate from the sick person or from the patient, culture it, purify it, and then introduce it into the healthy person, then the healthy person is becoming sick. The last postulate is that the microbe must be recoverable from an experimentally infected host. It means that the microbe should be able to the last postulate means that we should be able to isolate the microbe from the second infected person. There are some exceptions to Koch's postulates. There are, first, the microbes could not always be grown in the laboratory. For example, some bacteria like Mycobacteria leprae, Chlamydia species, and also viruses could not be grown in the laboratory in vitro. So we have to grow this kind of microbes in the live cells. The second exception is cofactors or genetic and immunologic factors in the host may play a vital part. And I think it is clear that the immune response is very important thing to defend the disease. The third exception is that the ethical consideration prevent applying Koch's postulate to diseases and pathogens that occur in humans only. You remember in the previous slide that to prove that a disease is an infectious disease, we have to introduce the microbe to healthy person, and then the healthy person has to get the same sign and symptom as the first patient. In some instances, this cannot be proven because the ethical consideration. We can prove it, of course, using animals, but not human. Some microbes are pathogen only for human. That's why we cannot introduce the pathogen, the dangerous pathogen, to human. So, as an example, in HIV viruses, which is pathogen only for human, we cannot prove the third postulate of Koch because it is unethical. The fourth exception is that the disease do not appear until many years after a specific infection. Again, the example of this exception is also the HIV, because we cannot prove the third and the fourth postulate in a short period. We have to wait until the disease is appear. Due to those exceptions, then we have to revise some of the Koch's postulate, and these are the guidelines to establish the cause of infectious disease. For the first Koch's postulate, the microbe must be present in every case of the disease. For the molecular Koch's postulate, the phenotype or property under investigation should be significantly associated with pathogenic strains of a species and not with non-pathogenic strains. And molecular guidelines for establishing microbial disease causation, the nucleic acid sequence of a putative pathogen should be present in most cases of an infectious disease. For the second postulate, the microbe must be isolated from the disease host and grown in pure culture. Specific inactivation of the gene or genes associated with the suspected virulence trait should lead to a measurable decrease in pathogenicity or virulence. And the molecular guidelines for that is that the nucleic acid sequence of a putative pathogen should be absent from most healthy controls. If the sequence is detected in healthy control, it should be present with a lower prevalence as compared with the patient with disease and in lower copy number.